All right, so using that little refresher about metabolism and exergonic reactions and endergonic reactions, we can talk about classification of microorganisms based on carbon and energy source. And this is something that we have already seen a lot as we were going through chapters 4 and 5, looking at the different types of organisms and how they were classified. But now we're going to actually get into the meat of these terms, what they're actually meaning. So they can be identified, our organisms can be identified according to the source of carbon that they use for metabolism, so the source of carbon they use for all these chemical reactions in the body, as well as their energy source, so how they get energy for these chemical reactions, for these anabolism reactions and catabolism reactions. So we have some prefixes here: auto, meaning self, meaning from the self, and hetero, meaning other or from other. And those refer to the origins of the carbon sources. Okay, so if an organism gets its carbon from its self, then it would have auto in the beginning of the term. If the organism gets carbon from somewhere else, from something else, then that would be hetero. So organisms that convert inorganic carbon dioxide, like CO2, into organic carbon compounds are called autotrophs. Okay, so troph meaning nutrients, or is supplied by, so nutrients or nutrition. So auto meaning self, and then troph for nutrition. So it's gaining its carbon source from itself. It's able to make it itself for its own nutrition. So this one is saying that an autotroph is one that can take inorganic carbon on its own, convert it into organic carbon. So plants and cyanobacteria are good examples, or well-known examples of autotrophs. So on the other hand, then we have heterotrophs. And heterotrophs, meaning other, so this is where we get our nutrition, trophs, nutritions or nutrients. Our carbon source is from other. So they rely on more complex organic carbon compounds as nutrients. So these are provided to them initially by autotrophs. So these would be many organisms, most organisms ranging from humans to many prokaryotes, as well as E. coli, are all heterotrophic. So if you think about humans, for example, you can think of us as being heterotrophs, meaning we are not able to take inorganic carbon, like carbon dioxide, and convert it into organic carbon. We are heterotrophs, meaning we rely on others. We rely on autotrophs, like plants, to do that. So a plant would be an autotroph. It converts CO2 in the air, converts it into a carbon source that is then stored within the plant, an organic carbon source. And then us as heterotrophs, we will eat the plant, and then that is other, so the plant is other, giving us nutrients, giving us our carbon source. So organisms can also be identified based on the energy source they use. So all energy is derived from our transfer of electrons. But the source of electrons differs between different types of organisms. So we have some more prefixes. Photo, meaning light, so getting electrons or getting energy from light. And chemo, which is referring to chemical, so getting our electrons or getting energy from chemical reactions. So those that get their energy for electron transfer from light are called phototrophs then. So phototrophs. So you can think of a phototroph would be a plant, for example. So they use light for their energy source, for their electrons. They're able to use the photons of light in order to provide nutrients. Whereas chemotrophs obtain energy for electron transfer by breaking chemical bonds. And so humans would be chemotrophs. So we do not gain it from light. We are not phototrophs. We can't just convert photons of light into energy. We have to get it from chemical bonds, and that's why we eat food, for example. And then we take food into our body and we break apart the food, and then that allows us to gain energy from that, so that the chemical bonds that are in food, we break those to get the electrons for transfer. And then further, we can break down our chemotrophs into organotrophs and litotrophs. So our organotrophs are, like us, humans, fungi, and many prokaryotes. We are chemotrophs that obtain energy from organic compounds. So we have to get it from things like plants. Whereas lithotrophs, the other type of chemotroph, so there's still chemical breakdown, so still breaking chemical bonds to get the electrons, but lithotrophs do it from things that are like rock. So litho meaning rock, so getting them from inorganic compounds. So we can break down chemotrophs, those that break down chemical bonds in order to get electrons or energy, into those that get it from organic compounds, organotrophs, and those that get it from inorganic compounds, which are lithotrophs, and those would be things like hydrogen sulfide or iron. And lithotrophy, just as a side note, is unique to the microbial world. So when we're talking about larger things like animals and plants, we do not see lithotrophs. So the strategies used to obtain both carbon and energy can then be combined in our classification of organisms, so according to nutritional type. So most organisms are chemoheterotrophs because they use organic molecules as both their electron and carbon sources. So chemo, meaning the breakdown of chemical bonds, and then hetero, meaning that we have to get it from other. So carbon sources come from other. Once we get those carbon sources, we break down the chemical bonds and then get the electrons and energy from that. So here is a nice summary of all of that information, classifications of organisms by energy and carbon source. So we have our chemotrophs, for example, those that break down chemical bonds to get energy. So here, that's why both of these under the chemotrophs are energy source, are chemical bonds that are broken down. And then we can classify further as chemoautotrophs, meaning the carbon comes from themselves, from inorganic places like carbon dioxide, or chemoheterotrophs. So again, chemo, meaning the energy source comes from the breakdown of chemical bonds, and then hetero, meaning the carbon comes from somewhere else, so from organic compounds, for example. So when we see, again, our chemoheterotrophs are the most common. That includes all animals. Most fungi, most protozoa, and most bacteria are chemoheterotrophs. Our chemoautotrophs then, so chemo from chemical bonds is where we get the energy, the electrons, and then auto, meaning they can get it from inorganic sources, are our hydrogen, sulfur, iron, nitrogen, and carbon monoxide oxidizing bacteria. Okay, so they're using these sources, right, hydrogen, sulfur, iron, nitrogen, and carbon monoxide, to get their carbon. So that's their inorganic carbon source. And then they use that, then they have a chemical breakdown within these bacteria to get their energy, the electrons. Then we have our phototrophs, photo meaning light. So here both of these have light as their energy source, so we're getting energy from the light. So then we have our photoautotrophs. So again, those that get their energy or electrons from light, photons. And auto meaning from self, meaning their carbon source can be made in themselves. They can get their organic carbon within themselves by taking it from inorganic carbon to start with. So then we have our photoheterotrophs, meaning photo, they get it from the light, right? They get their energy from the light. And then hetero, meaning they have to get the carbon as organic compounds from somewhere else. So our photoautotrophs would be all plants, right? Because all plants use photons of light from the sun for their energy, and then they use carbon dioxide as their carbon source mainly, which is an inorganic source of carbon. So our photoautotrophs are all plants, algae, cyanobacteria, and green and purple sulfur bacteria. Then we have our photoheterotrophs. Again, light, so using photons from the sun as their energy source, as their electrons, and then they have to use organic compounds for their carbon source from elsewhere. These would include the green and purple non-sulfur bacteria and heliobacteria. So non-sulfur, because of course sulfur would be our inorganic, which we see up here, which is why we see the green and purple sulfur bacteria appear as photoautotrophs. All right. So then we're going to take a little bit of a closer look at some of these electron interactions. So this likely will be familiar to you. We'll go through it again and then talk about it, how it relates to microbes. So if you recall, we have oxidation and reduction reactions in metabolism. So we transfer electrons, right, between molecules, and this is important because most of the energy is stored in atoms, and most of the energy stored in atoms is based on this transfer of electrons within molecules, and it's used to fuel cell functions. So we have this high energy electron situation where we have our different orbitals, our different electron shells in our atoms, and the way that atoms make bonds or break bonds is based on whether or not we have a sharing or a breaking apart of these electrons. So the transfer of energy in the form of electrons allows the cell to transfer and use energy incrementally. So in small packages. So transferring electrons from one molecule to another molecule is going to be a smaller package of energy rather than having some explosive burst that will then just be destructive. So using these little tiny packages of energy and electrons is a way that we're able to contain these reactions and use the energy within an organism. So as a reminder, reactions that remove electrons from donor molecules are called oxidation reactions. That leaves the molecule oxidized. Okay, so reactions that take away electrons, so if we are removing electrons from the molecule, so we have a molecule, we're going to take away an electron, then that is an oxidation reaction. And that leaves this molecule oxidized. Now the molecule that has the electron now that took it is reduced. Okay. Then we have the opposite. We have those that add electrons. So if we have our molecule and say we add an electron to it, they are those that add electrons to the acceptor molecules will leave them reduced. So now this molecule is reduced, and this is a reduction reaction. And that the molecule that gave away the electron has been oxidized. Okay, so basically when we lose an electron, it's oxidized. When we gain an electron, it's reduced. So when we have a situation where we add electrons to acceptor molecules, we leave the molecule reduced, and that's called a reduction reaction. In a situation or a reaction where we remove electrons from the donor molecules, that leaves them oxidized, and that's an oxidation reaction. So since we have electrons moving from one molecule to the other, our oxidation reactions are occurring at the same time. So as one is oxidized, the other is reduced, and then we have a transfer again, then the other one's oxidized, the new one's reduced. So these pairs of reactions are called oxidation-reduction reactions or redox reactions. So when we have these reactions, these redox reactions that I just briefly went through, you should already be familiar with from a previous biology class or chemistry class even, we talk about energy carriers. So within biology we have energy carriers that are going to go through and do these redox reactions. Our energy carriers are going to be NAD+, NADP+, FAD, and ATP. So the energy that is released from the breakdown of chemical bonds within nutrients can be stored either through the reduction of electron carriers or in the bonds of ATP, adenosine triphosphate. So in living systems, a small class of compounds function as mobile electron carriers. So hopefully you're familiar with these from previous classes. We have electron carriers. So they're basically, these guys up here, our electron carriers, are going to grab a hold of electrons and move them somewhere else in the cell. And then they will release those electrons. And that's a way for us to move energy around. So when we are breaking apart bonds, then we're going to be utilizing energy in that situation, meaning not using, but we'll be gaining that electron in that case. So when we have a more complex molecule that is broken down into its individual molecules, then we're going to have that spontaneous release of energy. Then we're going to grab that energy in the form of an electron, and then that electron carrier will carry that electron elsewhere, including with its energy, and then it will drop off that electron in another location that is going to be building a molecule, because remember, when we build a molecule, that requires energy to be put into it. Okay, so that's what we're going to see with these electron carriers. So these are molecules that bind to and shuttle high energy electrons between compounds and pathways. So the principal electron carriers we will consider come from the B vitamin group, and they are derivatives of nucleotides. So our NAD plus is our nicotinamide adenine dinucleotide, so NAD. Our nicotine adenine dinucleotide phosphate is our NADP, which is our second one up here. Our flavin adenine dinucleotide is our FAD, which is up here. Okay, so those are three principal electron carriers: NAD+, NADP+, and FAD. So these compounds can be easily reduced or oxidized. So our NAD plus, can then be NADH. So once it gains an electron, that's in the form of hydrogen here, we have our NAD+, we gain an electron giving us NADH, and then it can carry that to somewhere else, release the electron, and become NAD+ again. This NAD and NADH is our most common mobile electron carrier used in catabolism. Okay, so NAD+ is the oxidized form of the molecule. The NADH is the reduced form, because reduced means it has that electron, and you can see that because of the H here. Oxidized means it does not, NAD+. So our NADP, nicotine adenine dinucleotide phosphate, NADP+, is the oxidized form of an NAD+ variant. It contains an extra phosphate group, so that's what the P is here. So we have our NAD+, and then NADP+, just includes another phosphate group. Now, it is another important electron carrier that forms NADPH when it's reduced. So when it gains that electron, now it's NADPH. Then when we see our flavin adenine dinucleotide, which is FAD, that's our oxidized form, and then its reduced form is FADH2. So our NAD plus and our FAD are extensively used in energy extraction from sugars during catabolism. So when we have our long glucose molecules, not long glucose molecules, but when we have long molecules of many pieces of glucose, which in the human body, for example, is glycogen. We store glycogen in the liver. We have other organisms that store glycogen as well. But glycogen is many, many glucose molecules all attached to each other. When the body would like to take those, catabolize them, so take our complex molecule of glycogen and break them down into individual glucose molecules, or even take glucose molecules themselves and break them down into pieces, we will see a use of the NAD plus and the FAD here. And that's in, again, chemoheterotrophs, which, like humans, we're chemoheterotrophs, and we do extensively use this NAD plus and FAD in the breakdown of glucose, for example. Then we see NAD plus and NADPH play an important role in anabolic reactions, so the building up of complex molecules, and we see that also in photosynthesis. And I remember that NADP is in plants because of the P going along with photosynthesis. So plants and photosynthesis is NADP plus, versus humans, for example, are chemoheterotrophs, which use NAD plus. So collectively, our FADH2, our NADH, and NADPH are referred to as having reducing power due to their ability to donate electrons to various chemical reactions. So they are reduced, so they have reducing power because they can then reduce others, meaning transfer the electrons from their own molecule here to another molecule over here to reduce them. So a living cell must be able to handle the energy released during catabolism. So remember, we release energy in catabolism. So for example, glucose, we take a glucose molecule, we catabolize it, meaning take that complex glucose molecule, C in catabolism for complex, and then break it down into its individual pieces. In that process of catabolism, we release energy. So a living cell must be able to handle that energy in a way that enables the cell to store the energy safely and release it for use only when it needs to. So our living cells accomplish this by using adenosine triphosphate, which is ATP. ATP is often called the energy currency of the cell. It's very versatile, and we can use it to fill any energy need within the cell. So our ATP, at the most basic, is adenosine monophosphate, or AMP. So adenosine includes an adenosine molecule. So adenosine molecule bonded to a ribose molecule, and then a single phosphate group. So that's where we get the mono part. Mono meaning one, and then phosphate. So adenosine, and then one phosphate group. And then both of those are attached to a ribose for the backbone. So our ribose is our 5-carbon sugar, and we find it in RNA. And then AMP, our adenosine monophosphate, is actually one of the nucleotides in RNA. So when we translate things or transcribe things, we make AMP is part of our RNA molecule, our mRNA. So the addition of a second phosphate then in this core molecule results in adenosine diphosphate, or ADP. So di meaning two. So our adenosine group attached to the ribose, also attached to the ribose sugar are two phosphate groups, diphosphate. So then when we have the addition of the third phosphate group, we form ATP, because remember up here it's adenosine triphosphate, tri meaning three, and then phosphate meaning phosphate groups to that ribose. So adding a phosphate group to a molecule in a process called phosphorylation, so adding phosphate is called phosphorylation, requires energy. So to go from AMP to ADP, we have to put energy in. And then going from ADP to ATP, we have to put energy in. Okay? Also, these phosphate groups are negatively charged. So negatively charged, and then they repel each other. And so when they are arranged, when they're attached to each other in ADP and ATP, they're repulsing each other. So there's a repulsion between them. So it makes them very unstable. They're inherently unstable because they're repelling each other, because they're negatively charged and they're very close to each other. So we had to put energy in to force these two negative phosphate groups to be right next to each other. And even though we put that energy in, they're very, very unstable. So the bonds between the phosphate groups, one in the ADP and two in ATP, are called high energy phosphate bonds because there's a lot of energy built up in there. We had to put a lot of energy into it to force them to stay next to each other. And so they're just looking for a chance to burst apart. And when these high energy bonds are broken, when they do break apart, when they release one phosphate, it's called an inorganic phosphate, or a PI, or they can release two connected phosphate groups, and that's pyrophosphate, or PPI, from ATP, and that's called dephosphorylation. So phosphorylation is adding the phosphate groups on, which requires a lot of energy and then makes a very unstable ATP molecule in the end. And then when we release those groups, break those groups off, that's called dephosphorylation. And in that case, energy is then going to be released and then it's going to drive these endergonic reactions. So remember, endergonic reactions are those that require energy. So the energy that's released from this ATP molecule can then go on to do endergonic reactions, which would be a building up of molecules, which would be an anabolic type of reaction. So if we want to look at that in an illustration, we can put in energy here. So here, let's look at this molecule here. Right here, this is our ribose sugar. It's 5-sided. You can see here, we have 5 corners. That's our ribose sugar. And then this is our adenosine. And then these are our phosphate groups. So there's 1 and there's 2 already attached, which gives us our ADP. And then what we're going to do is add another phosphate group on here. You can see here they're all attached to each other, or will be. And so then forcing those negative phosphates next to each other takes a lot of energy. We can put energy into this, either solar energy from the sun for a phototroph, or a chemotroph using chemical energy. Can then put energy in. It would be then undergoing phosphorylation, because we're going to take a phosphate group and add it. And when we do that, we come up here and we get ATP, adenosine triphosphate, tri meaning 3. So here's our adenosine group, our ribose group, and then our triphosphates, or 3 phosphates. Now remember, these bonds between the phosphate groups are super high energy because these phosphates don't want to be next to each other. So they're very unstable and they've got a lot of energy built up. When we go through the process of dephosphorylation then, which would be cleaving these phosphate groups off, then that releases all of that pent-up energy in those phosphate groups. And then we can use that to do cellular work. We can do that for anabolism, or some anabolic processes. So we can take that energy and put it into other reactions, endergonic reactions, those that require energy. So then down here, we can see something similar when we're talking about exergonic reactions and endergonic reactions. So we have our ADP, which we just saw. I could probably get both of these in the same situation here, same screen. Our ADP, which only has the 2 phosphate groups, and we're going to add an inorganic phosphate, the PI. When we do that, that requires energy. So this is an endergonic reaction here. It requires energy to go into it to force this inorganic phosphate to then become attached to the other phosphate in here. We get that energy from a process here. So this is an exergonic process. We're taking glucose and then we are breaking it down, and in that breaking down process, in catabolism, we are releasing energy. When we release energy, we can put it into this creation of ADP into ATP. So we take the energy from catabolism, our exergonic reaction, those that release energy, put it into ATP. This is an exergonic reaction. So our exergonic reaction here is going to fuel our endergonic reaction over here. Then we have ATP, which is storing energy for us. If we want to then, say, take glucose and make a polysaccharide, poly meaning many, saccharide sugars, like glycogen, as I mentioned, glycogen. If we want to make glycogen, which is many glucose molecules, polysaccharide, we want to form bonds between glucose molecules. Well, if we're forming bonds, that means we're going to have to put energy into it. It's an endergonic reaction. Where are we going to get that energy? We're going to get it from ATP. So ATP is going to go through dephosphorylation, taking off a phosphate group. That is an exergonic reaction, releasing energy. That energy is going to go into our endergonic reaction here, which is our bond formation, making our polysaccharide. So we have phosphorylation down here, dephosphorylation here, and then we have that movement of energy between our exergonic reactions and our endergonic reactions. And that's very, very important for understanding these processes that the microbes, and even not even microbes, but humans like ourselves and plants and things go through in order to move energy around the cell in order to build up and break down. All of the things of metabolism, all of the processes of metabolism.